Okay, so I shortened this talk just a little bit because I knew the first talk would go a little bit long. Um, but the way I shortened it was I just hid the slides, so from presentation. But so if you download the talk, you can actually get all the slides for reference if you want. So anyway, um, okay, so now just to jump into the next part. Okay. So looking at functional MRI contrast and limits of spatial and temporal resolution. So I'm going to start out with functional contrast and then talk about the limits of spatial and temporal resolution and talk about all the things that could limit in spatial and temporal resolution. So the functional contrast in MRI can be divided. I mean, these are all the hypothetical contrasts that you can actually see with fMRI. Everything from blood volume, the first fMRI scan was with gadolinium, bold contrast, perfusion. By looking at, I'll get into it, by looking at the ratio of bold to perfusion with the very specific types of modulations, you can actually look at changes in CMRO2, metabolic rate. You can also non-invasively look at blood volume changes with fMRI. These last three are a little bit more experimental. They're probably, some of them may never happen, and maybe all of them may never happen. But there's a certain number of people working on trying to look at neural currents directly with fMRI. There's looking at people trying to look at very subtle diffusion coefficient changes, inferring maybe cell swelling with brain activation. It's a non-bold sort of contrast, potentially. And also temperature changes. You can see that by subtle phase shifts. Okay, so starting with gadolinium. As I mentioned, some of these are going to be overlapping, so I'll go over relatively quickly the ones that were. So with gadolinium, this is the first fMRI experiment where you had, as I mentioned before, an injection with the equiplanar time series with gadolinium. It takes about 10 seconds to reach the brain, passes through. As it passes through, it's paramagnetic. It attenuates the signal, causing dephasing. As it washes out, the signal returns back to normal. You integrate under this curve on a voxelwise basis to get the blood volume during rest, during activation. And so just to reemphasize, this technique is actually used a lot clinically just to make maps such as this. This resting state blood volume map is extremely informative for stroke, for other sort of disorders. Okay. But not only that, also for looking at this longer edge of the curve, you can actually start to look at T1 effects, which I won't go into. But so gadolinium in general is useful, not for brain activation though. So this was the one of the one and only, I think, published brain activation maps using blood volume contrast with gadolinium. And as I mentioned, it's based on the fact that when the gadolinium passes through the bloodstream, just like with deoxygenated blood, it causes field distortions. And where there's larger magnetic fields, the spins are processing faster. Lower magnetic fields are processing slower. They dephase. That's what causes the signal decrease. Okay, and this is just to still kind of illustrate this point. When it passes through, you might have a vessel that looks like that, and then as it washes out, it looks like that again. So you have that sort of effect. Okay, bold contrast. I talked about this before. And this is sort of going into one aspect of bold that I didn't talk about. And that is, so when we do MRI, when we do a T2 star weighted signal, we essentially give our RF pulse and we look at the transverse magnetization as it's decaying right here. And so typically in the brain, the T2 star, happens to be at maybe 1.5 Tesla, is about, the resting one is maybe 48 milliseconds, and during activation, it changes by about 2 milliseconds. And you have this very subtle shift in this exponential decay. So here's an important point, though, and I don't know if you can see this. Oh, yeah, you can. You can see it much better on the screen than my computer, is that if you take the difference between these curves, you get a peak at about the echo time equal to the T2 star. And that's why we collect at the T2 stars, at the TEs that we do. We don't go any longer because this difference starts to attenuate as the overall signal starts to drop. Basically looking at the difference between two exponentials, the peak is always at the time constant that defines the exponential curve. And this is why, even though the percent signal change keeps on going up with echo time, it can keep on going up forever, the contrast that we get is really because of this peak right here, the difference in the signal. And this is actually showing that a difference between 1.5 and 3, could easily be 3 versus 7 Tesla. And the contrast is not only dependent upon this change in T2 star, but it's also dependent upon the baseline T2 star. And that's why it's really important to get a good shim at the base of the brain. So for instance, if you're looking at 3 Tesla, for instance, and you have maybe a meaty part of cortex that's it's shimmed and nice and homogeneous, you might have a T2 star that is 60 milliseconds. So your peak, your curve would be here, and your contrast is here. But if you're actually at a part in the base of the brain where you have a signal dropout near the ears and the sinuses, you might have a T2 star that is down here at about 10 to 20 milliseconds. And so the maximum contrast that you can get is much less than up here. And if you're only collecting at a TE of optimizing this contrast, you won't see anything down here where the T2 star is low. So you'll have no signal where you're looking, and the contrast will be lower. And that's why actually maybe 1.5 Tesla might even be good for looking at the base of the brain, because it's easier to get a better shim at the base of the brain. And even though while the overall contrasts are less than the 3 Tesla, you might have less of this sort of effect at the base of the brain. Okay, but still, if you're at the optimal echo time and the T2 star, the echo time matches the T2 star of predominant tissue, yes, the T2 star goes down a little bit at 3 Tesla, but not that much. And so overall, the change in contrast, the contrast gain is much higher at 3 Tesla. And it corresponds linearly, approximately, with field strength. Okay, a quick comparison of spin echo versus gradient echo. And historically, this was something that was interesting because people were trying to get at higher and higher spatial resolution. They always worried about draining vein effects. Gradient echo is great for being sensitive to the signal, but because you have large vessels that are downstream from the areas of activation that have a large penumbra, they might enhance the signal overall, but they increase the amount of uncertainty as to where the true activation is coming from. So people were trying various techniques. So the concept here is that if you apply a 90-degree pulse, the signal will decay really quickly. This is the gradient echo that we look at. If you apply a 180-degree pulse, it will basically flip the spins, and they'll refocus at some time exactly double the time away from where the 90-degree pulse was. And that amplitude of that signal is proportional to the T2 of the signal. And that is less sensitive to various signal dropouts as well, like macroscopic large dropouts at the base of the brain. They all become refocused, or mostly refocused, by the 180. So there's an advantage there as well. And that is T2s are generally about 100 milliseconds, T2 stars is about 30 milliseconds. Okay, and here basically, once again, relying on the fact that all we're looking at is the constructive and destructive addition of diffusing spins through a gradient. And this is I'm going to try my best to try to say this clearly, but essentially, spin echo is sensitive to small compartments, and here's why. So one thing I didn't talk about, if you have a compartment that is, let's say, 20 microns, which is mid-sized compartment, you might have a spin that's maybe here, located right here when you give the first RF pulse. But that spin during the time before you collect the data might diffuse a certain distance such that it's experiencing a completely different gradient. So that's great for gradient echo. It just dephases everything. That's fine. But for spin echo, that spin that's now experiencing this different field will be processing at a different frequency, so they will never become refocused. Okay? So if the compartment is super small, then the spin will diffuse right by that gradient and not get dephased and still have the same frequency. If the compartment is super large, the spin won't be moving that much and it'll be perfectly rephased. So the idea here then is that essentially spin echo sequences are sensitive to the spatial scale that about matches the distance that a spin diffuses in the time that you're looking, which is on the order of maybe 5 microns or so, 2.5 microns to maybe 5 microns, which is the sweet spot for spin echo sequences. Whereas gradient echo, the bigger the compartment, the less you have diffusion blurring things out, and the better off you are. Whereas with spin echo, so the more it's attenuated here, the more sensitive you are to bold contrast. So the less attenuated, the less sensitive you are. So with spin echo, if you have a massive compartment, it will completely refocus almost everything, even if it is diffusing, you won't see anything with spin echo. With a compartment that's too small, you also won't see anything. That's called the fast exchange regime. But this mid-range, 5 microns, you'll see some attenuation. It will still be less than gradient echo, but it'll be there. So this sort of summarizes what I just tried to say right here, is that spin echo sequences have a sweet spot for the compartment size of the susceptibility perturber. That's from 3 to 15 microns, whereas gradient echo just goes up and stays up. And so that's why you might be more sensitive to small vessels. And here's a quick comparison. This is a bolus injection of gadolinium, and we had this unique sequence in which we simultaneously collected gradient echo and spin echo data. And you can see these maps. They're nicely illustrate that with gradient echo, you see these large blotches where it's most sensitive to large vessels. You have the sinuses and the big vessels, things like that. Whereas spin echo, it's much more of a small vessel contrast right here. Okay. And basically, the way you can look at it, though, is that the brain, the vasculature has not only big and small vessels, but you have red blood cells inside of it. And unfortunately, the red blood cells are small compartments. And so you might have a massive vessel, but you have a ton of small compartments here, so spin echo still sees those. And so that's one of the big shortcomings of spin echo, is that you still see, it's still sensitive to all these small compartments inside the vessel. So as a quick comparison here, this is an image. This is where the dark areas are where the vessels are. And you can see actually gradient echo shows more contrast. Spin echo also shows a large vessel effect, though, right here. So it's not, and this is only at 1.5 Tesla, where there's more intravascular signal. The higher field strength you go, the less of this intravascular signal you go. So maybe the benefits of spin echo might be manifest more at higher field strengths. And this is what this sort of illustration shows in a sort of busy way. So basically, this is the intravascular contribution as a function of echo time for spin echo versus gradient echo. And essentially, this is actually also across field strengths from 0.5 to 4 Tesla, 0.5 to 4 Tesla. And you have the higher field strength, the more the signal is gone intravascularly. And one other added benefit of this, the fact that blood T2 star is much less than that of tissue at higher field strengths, is that you can make these wonderful venograms. And basically showing that the signal inside the vessels is already completely dark at a typical echo time for gradient echo imaging. So you actually see these, all the veins showing up as dark vessels, kind of like Ogawa's original plot that I showed you. And actually, Ravi Menon first showed this as well, when he showed basically this is at 4 Tesla, he points his arrows to all these dark dots, which are basically large vessels, that he shows that this is also where the hot spots of the gradient echo signal is. And here's Mark Hakey actually pioneered this technique. He was one of the pioneers of this technique. Here showing an early paper of what he coined as susceptibility weighted imaging. This is some work by David Norris. Also a reverse contrast showing the vasculature. And this is some work by from our group. Also, Jeff Dunn's group has been pioneering susceptibility contrast as well. And this is showing that. And that is, so basically these are nice venograms. They become nicer at higher field strengths. There's other ways of even making them better, but I'm not going to get into that. Essentially, just looking at the T2 star contrast is mostly good enough. So it turns out phase information, this is from Peter Van Gelderen and Jeff Dunn, phase information also reveals a unique type of contrast that just doesn't show up with magnitude, that will highlight blood volume and also iron deposits and other types of things like that in the brain as well, that you won't get through the magnitude. So here's a beautiful example of basically showing magnitude versus phase. And you can see in the magnitude images, and so I'm not going to get into the NMR phase, but essentially it's a way of reconstructing the images that that you just don't get when you reconstruct the magnitude image. I'm not going to go into the physics right now, but essentially you do see a lot of types of contrast that's due to susceptibility contrast that you just do not see in the magnitude. So that just illustrates at high resolution. It's illustrated at high resolution. This is at 7 Tesla right here. Okay, Heikki also tried his best to do oxygen quantification with bold contrasts. So it turns out that you can actually, if you make some assumptions about vessel orientation, vessel geometry, you can infer based on what the T2 star of the blood is, what the oxygenation is. And so he did his best at that, and he actually came up with a figure that was approximately what is typically measured as venous oxygenation, about 50%, 55% oxygen saturation. This isn't used that much, but it's a technique that potentially can get at oxygenation. Other techniques have been advanced. This is a technique that does a combined, sort of pioneered by Hanzheng Liu, where it combines arterial spin labeling and multi-echo imaging. So essentially, you're just looking at the vessels, and then you're measuring the T2 star within the vessels that you highlight with arterial spin labeling. So it's essentially it's a way of quantifying oxygenation. It's called trust MRI in the vessels. And so with the right assumptions and with the right models, you can make all types of parameters, maps of other types of parameters by looking at, for instance, the ratio of spin echo to gradient echo. If you know what their sensitivities are, as we do, you can get a vessel radius map. You can get other changes as well. You can start to get at then venous blood volume, venous density, and then the radius maps right here. So you can get all kinds of rich vascular information just looking at the comparison between spin echo and gradient echo. So a quick summary between spin echo and gradient echo is that you might have increased specificity where the intravascular signal is low for spin echo. You have less sensitivity to rapidly flowing blood because it's excited by the 90, it flows out of where the 180 hits it, and the 180 hits then the tissue that hasn't flowed that much, and so you have less sensitivity to inflow effects, and then less signal dropout because you refocus all these macroscopic susceptibility effects. Problem is you have less slices per TR, you have much lower functional contrast and noise by a factor of 2 to 4. The acquisition window is still wide for echoplanar imaging, so you still have a lot of T2 star weighting, and you have a large intravascular signal. At most, it's even present even at 7 Tesla. It's not large at 7 Tesla, but it's there. So it still sort of clouds the interpretation of it. So I would only use spin echo at 7 Tesla if I really, really, really want to get at high resolution. Otherwise, maybe not. And actually at 7 Tesla, if I really want to get at columns or layers, I might now, given Laurentius Huber's work, I'm convinced that vaso is much more sensitive to capillary effects, more so than even spin echo echoplanar imaging. So I would probably use vaso, which is a blood volume sensitive technique. Okay, so if you want to remove the intravascular signal, one way to do it is adding diffusion weighting. So diffusion weighting is mostly attenuates spins that are diffusing based on the fact that if you apply gradient and they accumulate a phase, you apply the opposite gradient, it will never become rephased because it's a different location. So essentially, this works for slow flow as well. So if you apply a little bit of diffusion weighting to flowing blood, it gets rid of the intravascular signal. So this is some early experiments where at 1.5 Tesla, so adding a very small amount of diffusion weighting attenuated the signal by 2/3. And here's the maps. B equals 0. This is a measure of diffusion weighting. B all the way up to 160. You have almost no contrast left. Okay. So that's, if you really want to get so the one experiment that people have tried, including myself, is to apply diffusion weighting and apply spin echo at 3 Tesla. And if you do that, you get absolutely no bold contrast whatsoever. You've gotten rid of everything, it seems. And so, unfortunately, those capillary effects that the spin echo still might be sensitive to are just not enough to really see much, at least at 3 Tesla. Okay, so this is sort of a complicated figure, but it gets at some of the aspects of the hemodynamic specificity. So you have, if you're looking, everything that's filled in is what the sequence is sensitive to. On the left, you have perfusion. On the right, you have bold. And here you have a time delay. If you're interested in temporal resolution, this is sort of a time spread from where you have perfusion out to the draining veins. This can be up to 4 seconds. So with perfusion, you're mostly sensitive to this part of the vasculature. You can be sensitive to the veins as well if you're not careful. If you apply velocity nulling, though, you are mostly sensitive to the capillary perfusion right here. So this is with no velocity nulling. This is with velocity nulling. With great echo, velocity nulling, applying diffusion gradients, you get rid of the intravascular signal, but maybe you might have the capillary signal left for both gradient echo and spin echo. So this is a chart that might be worth more studying afterwards without getting into too much detail. I realize I'm almost starting to run low in time. Okay, looking at perfusion. As I mentioned, it was pioneered by Alan Kretsky and Detra and Williams in Pittsburgh, but it's been going on here. Epistar was by Bob Edelman. FAIR was developed by Sanji Kim. And as I mentioned before, with Epistar, you have a tag below the plane that you're interested in. You can apply a tag above as well. There's many different other versions of this, but essentially you apply these pairs and then you subtract the pairs and you only get that which is flown into the plane. And as I mentioned in the previous lecture, the longer you wait, the more you get. You start out with, if you only wait 200 milliseconds, you get something that looks like angiography, the only most rapidly flowing blood. If you wait a second, you get something that looks like perfusion. And these are some more recent perfusion images that are obtained. It's still not used clinically that much because the contrast to noise is still lower than if you were injecting gadolinium to get perfusion or blood volume. Here's some of the pioneering papers, though. Williams, Edelman, Kim, and Kwong. Okay, and here's a comparison between perfusion contrast and bold. This is kind of, in a nutshell, what it shows. So bold, this image was obtained in about 3 minutes. With perfusion, you get rest and activation, but this took about 7 minutes to obtain, and it's still a lot noisier. So that's kind of sort of a comparison. If you really want to go to high resolution, you might want to do perfusion, but you're really giving up a lot in terms of contrast and noise. But you can get this baseline information, which is potentially useful. And here's just basically a high resolution of bold and perfusion sequences. And you can see that the hotspot with perfusion is probably more localized to capillaries and to areas of where the true activation was, whereas bold, you might get these large vessel effects. Right here. It's a very, you have a shift of the center of mass by up to 1 centimeter between perfusion and bold. But for the most part, that doesn't really matter that much. For most systems level mapping, it doesn't really make a difference. People are always struggling to get as much contrast as possible. But here is one nice study where it's actually potentially really useful to do perfusion imaging, and that is for very slow activations. So if you're looking at, so this is basically a plot of relative contrast, functional contrast versus on-off cycle. So one of the reasons why we do, with bold contrast, why we did block designs that were like 10 seconds on, 10 seconds off, and so on, or else did event-related, rapid on, multiple event-related, is because there's this constant drift in the baseline. And there's ways of correcting the drift, but you're never certain that you have it fully corrected. And so bold contrast, actually, if you give a 3-minute scan, 3 minute on, 3 minute off, you'll be contaminated by a lot of the drift in the baseline drift in the signal. So that's why we don't do bold that way. And if you did it that way, the relative contrast of bold would go way down. Turns out, I'm sorry, this is the frequency. So very low frequencies versus very high frequencies. And high frequencies, of course, you just lose amplitude of the signal. But and bold is much better than perfusion for most cases. But at very, very, very low frequencies, this is a nice paper by Aguirre in 2002, shows that perfusion beats bold if you go to super low frequencies. So here is their map that basically showed your standard. It's worthwhile to compare the optimized bold with ASL perfusion right here. Nice, better contrast for the most part. And then if you go to about 4 minutes on and 4 minutes off, bold is gone, whereas you still have perfusion. And 8 minutes, bold's gone. Perfusion, this is even an hour on, an hour off. You have a nice perfusion map. And even at 24 hours. So this is where you scanned a person once, one day, and then brought them back the next day, scanned them. You have, if you obviously, same person is registered. Perfusion is a nice self-calibrating technique that the baseline is stable. So, water. So if you were doing an experiment like this, you can imagine slow brain activation with like state changes, with like looking at drug effects or looking at, effects with eating or things like that. You can imagine perfusion would lend itself to that. And also it might be worthwhile to look at perfusion as a sort of a metric of baseline metabolic rate. Like you might be able to collect maps of perfusion of maybe people with ADHD versus maps of perfusion with those who don't, and just subtract the maps to look at the areas that might have chronically elevated or decreased perfusion as sort of maybe a metric of what the ongoing activity is of those tissues. So I'm going to skip that. Okay, CMRO2. So the way you can extract CMRO2, which is cerebral metabolic rate of oxygen in the brain using fMRI, is by using the understanding that if you give with bold contrast. So for instance, if you give a task, you have an increase of perfusion in the area. You also have a slight increase in metabolic rate, which actually blunts the bold signal just a little bit. It pulls it down just a little bit. But if you were to have a global increase in flow without any brain activation, which you can induce by giving a person carbon dioxide or having them hold their breath, then you won't have that signal blunted. So by any increase in metabolic rate. So if you look at the ratio of flow to bold effects with giving CO2 versus giving an activation, then you'll have different ratios because with activation, the metabolic rate blunts the bold relative to perfusion. And so that's what Rick Hoge did here. This is a nice work in 1999. Tim Davis did it just before him, but this was a nice study that sort of illustrates the facts. So he did an experiment in which he was measuring CBF, and the green line is visual stimulation, was one experiment. The white line, he actually somehow managed to map. This was the amazing part. He mapped the amplitude with a graded hypercapnia experiment, and he was able to match the signal changes. So in bold, though, if you give visual stimulation, this is looking at the visual cortex. Notice the bold signal goes up, but it's blunted relative to if you give just pure CO2, a global flow change, because there's no increase in metabolic rate to blunt it, to decrease it. So this signal difference, essentially, the difference between this signal and this signal is purely due to the brain pulling out oxygen from the blood when it's becoming more active. So essentially looking at the difference between these two signals, he was able to make a quantitative map of CMRO2 in the brain. So this is a quantitative CMRO2 change map. We're still working on better ways. There's still ways now with a certain number of assumptions of mapping out baseline CMRO2 as well, which would actually sort of push fMRI into the realm of PET, which could be useful, or at least one aspect of PET. Okay, so blood volume imaging. So this is a non-invasive technique for looking at blood volume. It was developed by Han-Zeng Liu at Hopkins when he was a graduate student. And the idea here is that if you give an RF pulse, if you give a inversion recovery pulse, it turns out that different tissue has different longitudinal relaxation rates, and blood has a different longitudinal relaxation rate than tissue. And it turns out that if you wait just the right amount of time as the signal is recovering, so when you give an inversion pulse, instead of giving a 90-degree pulse where it takes the magnetization to 0, you give an inversion pulse, it flips it completely. It'll recover back based on T1. And at some point, the blood signal will go through 0, which is the null point. At that point, you have a little bit of tissue signal because the tissue T1 is such that it's a little bit faster, so it would be recovering faster. And then here's where you start your imaging experiment. You apply your 90-degree pulse here, and then do your imaging. So if you do that, you can actually image blood volume, because now you have this void of signal where the blood is. You have 0 signal where the blood is. If you have an increase in blood volume, you have more of that 0 signal, which will cause the signal to go down, which will cause overall the signal to go down, especially if your TE is short enough. And so here's the basic BOLD experiment where the signal goes up with visual stimulation. If you apply vaso, the signal goes down with stimulation. So it's a really nice technique. And the contrast to noise is probably a little bit less than BOLD, but I would say this: if you really care about columnar or layer-dependent activity, I would say that, as I showed the results in the previous lecture, I would say that the specificity is at least better than perfusion in terms of looking at capillary blood volume changes. It turns out most of the interesting blood volume changes, if you look close enough, change, at least in my opinion, it seems like they change at the level of capillaries. Okay, now with the last 20 minutes, I'm going to zip through this very quickly. Not too many slides, but it's conceptually a little bit easier to digest as well. So once again, I'll just skip this right here and go right into spatial resolution. So first of all, you're limited by spatial resolution in echoplanar imaging. In the imaging technique itself, you're limited by the T2 star decay. As I mentioned, there's only so much time you have to collect your data. Your T2 star is typically about 60 milliseconds or 70 milliseconds at best. So you only have that much time. So, and also you're limited by your hemodynamics. And right now we're sort of at the point where we can collect high resolution, and yet we're still have a problem in terms of exactly what part of the vasculature is showing you want to get the vasculature that is closest to the neural activation. But if you have neural activation, let's say here, you might have it drain into a vein, and then that might drain downstream into a sinus. And so you might see BOLD changes that go all the way along this draining vein. So we want to make sure we don't see that, and as I was talking about. And as I showed you, there's columnar activation. People have tried to quantify the point spread function. Most recently, Amir Shmuel in 2007, at 7 Tesla, it's a little bit better because you're more sensitive to capillary effects. It's 2.3 millimeters. As I mentioned as well, the Tour de Force was doing orientation columns, 2008. This hasn't been replicated, but it's still something that was super impressive. That is sort of the demonstration of the highest possible resolution. This is on the order of 0.5 millimeter resolution in terms of the functional resolution. As I mentioned, Laurentius Huber has sort of been pushing that envelope of looking at layer-dependent specificity, and this is, once again, finger tapping versus not tapping. You have specific inputs from sensory cortex that are modulated by that modulation, and by taking the difference between these two curves, you actually get, with a blood volume change, a very clear change in layer 2 and 3 of the motor cortex. And as I showed, these columns could be almost everywhere. So temporal resolution. So let's start at the other extreme. So very slow changes. This was a long time ago. There was a controversy of, does the signal actually, in the early days in BOLD, there was a lot of fun in terms of trying to answer these questions. There were some groups that said, oh, the BOLD signal habituates. So you can't give a stimulus more than 3 minutes because the hemodynamics will habituate. Turns out that actually ended up being neuronal habituation that occurred. And it actually seems that as long as you keep the brain going, this is an experiment where we basically tortured our subjects. We did 20 minutes of continuous visual stimulation flanked by 2 1-minute stimulations just to correct the baseline. Basically, the BOLD signal stays up the entire time. So it's very clear that the hemodynamic coupling is high fidelity, sort of related to ongoing neural activation. Okay, how brief can you go? So this is, as I mentioned, the paper by Blamir. He went down to 3 seconds. This is 92. You can go down much lower. So this is some unpublished data showing going down to 250 milliseconds. What happens, though, is that things become slightly nonlinear the lower you go. So it's a linear function of duration within this period between about a second to like 5 seconds. But below that, the hemodynamic response seems to saturate at this low level right here. You get more bang for your buck, essentially. At 250 milliseconds, the signal looks almost the same as at 500 milliseconds. Another way of thinking of temporal resolution is how fast can you turn on and off the brain and still see a signal. This was an old experiment which we went from 24 seconds on and 24 seconds off down to 1 second on and 1 second off. You can see two things happen. One, the amplitude starts to be, and this is a summary of the amplitude of this effect. The amplitude starts to quickly drop off at about a 6 second on, 6 second off paradigm. But here it looks almost exactly like a sine wave. So if you're doing like, some pure Fourier analysis of your data, you might want to activate at this frequency. Another thing happens is that typically the signal becomes saturated in the on state, which means that it takes a little bit longer to return the baseline. So there's generally. So it becomes saturated in the on state. And just recently, this was actually from last year, John Polomini at MGH and his group are demonstrating that unlike what you would get from a predicted response, you can actually elicit with enough averaging and looking at just the right type of stimulus, V1 can be reliably detected up to 0.5 hertz stimulation frequency. And just this year, he actually went up to 0.75 hertz frequency. And I think you can get up to 1 hertz. I mean, I think that, maybe even looking at this with the right amount of averaging, you can actually see something. But we'll see. But I think, so the hemodynamic response, and it's interesting though too, because this fast response is there, but it doesn't match your typical model, your typical gamma variant of the hemodynamic response. That would never see that. So there has to be some higher frequencies that are here that we haven't quite been able to capture yet. I mean, it's a very low amplitude response, so it might be something like this, but it's just a little bit higher off. And this could be more of a blood volume effect than anything, who knows? Okay. All right, temporal, another way of thinking about the way that people typically think of temporal resolution with fMRI or with brain mapping is how well can you resolve one part of the brain turning on versus another part of the brain. And that's what people really think about when they think of temporal resolution. And that with fMRI, unfortunately, is a mess. And because of the fact that you have your hemodynamic, your plumbing that sort of drains through. And so this kind of illustrates that point. You have, once again, this is simultaneous finger tapping. This is just bilateral finger tapping. And if we were mapping the magnitude, we see the largest magnitude effect where the large veins are right here. This is a dark dot in the venogram where the large vein is. That actually corresponds to where the largest latency is. So if you have the longest latencies, it's typically, and the way we actually mapped latency here, so you can give a very brief response and still elicit a response. So you can give all the way down to, like I mentioned, 500 milliseconds. This happened to be a 1 second stimulus. What we did here is sort of a poor man's way of measuring latency. You took this curve and you shifted it along and found the maximum correlation at where this curve was shifted. AFNI can do this pretty easily. And then map out whatever that shift was that optimized that latency and that optimized that correlation. And so that's how we made this map right here. So, but if you look at this distribution, there's a distribution of latencies of -2 seconds to +2 seconds. And that's all pretty much in an area of the brain we know is like synchronized, it's firing together. So that's because of the hemodynamics. That's because you have upstream effects and you have downstream effects of the blood flow. And so that really is a problem. One way of potentially getting around that, and it's not really solving that problem, but it turns out that the hemodynamic response, while it's messy over space, it's very well behaved in each voxel. So that means that if you have a control experiment, if you modulate the timing and then subtract those modulations, you can actually get at some very subtle timing shifts. So here's an example of staring at a fixation point, first staring at a left visual checkerboard for 500 milliseconds, and then staring at both, and then the left one goes down, and then the right one. So, and then looking at regions of interest from the left and right hemifields. So basically one hemisphere will activate the right visual cortex, one will activate the left. And what you see if you pick your regions of interest just right, and this is, I just want to show this because it's kind of what people tend to do. I mean, if you pick your region of interest just right, you can see a latency difference, and that's great. But in fact, that's not how things actually look. If you actually look at the data, at the map itself, it's not clear how to choose your region of interest. In general, this side might look like it has a, these are maps of latency. This is plus or minus 2.5 seconds. It might look a little bit like this is happening before this, but it's not, there's so much spread, it's hard to see. So yeah, if you picked your region of interest just right, if you just made sure you had the same number of vessels on each side, then you might be able to get that. Okay, so what we did though is we modulated the experiment. We actually had, so this is a 500 millisecond delay from right hemifield to left hemifield. And then we had then we flipped the order. We had a 500 millisecond delay between left hemifield versus right hemifield. And just eyeballing these, they don't look that different, but if you subtract these maps, you get a really nice map of left and right visual cortex. So the basic idea here is that, yes, if you modulate the stimulus and see what part of the brain changes with the modulation and subtract those, you can actually get at a higher temporal resolution. And we've done the same thing down to 250 milliseconds, and we get, it's not quite as nice of a map, but we get a difference. So you can get relative modulations of timing that go much faster. And there's all kinds of things that could happen. So you could actually, neural activity can cause, like for instance, if you, other ways of picking apart the signal as opposed to just looking at latency, you could look at how the width changes with neuromodulation. That might be a bottleneck in processing, or like an upstream effect. Or you can have the latency shift, which if this is a latency shift with a neural modulation. So you're kind of subtracting out the hemodynamic effect. And if you have that, that could be a downstream effect. So here's just one quick example of an experiment that we did that kind of tried to push that proof of concept. And that was essentially if you look at either words versus non-words and you respond, you make a judgment, whether it's a word or non-word, it takes a little bit longer to make a judgment that something is a non-word. And if you look at words that are rotated, it turns out that the amount of time it takes you to respond is exactly proportional to the angle of rotation. So what we did an experiment which we varied the latency of the response in two different ways. One was modulating from word to non-word, and the other one was modulating the angle of rotation. And if we're looking at one specific part of the brain, Broca's area, the left anterior prefrontal gyrus, we find that if we're just looking at the latency here, we find that with the word versus non-word, the onset doesn't change, but it seems like the width changes. And there's other changes though too that we don't completely understand, but it seems like the width might change. And that might imply that this area, when you're looking at the word versus non-word, this is the bottleneck of the processing. This is where the word versus non-word discrimination might be happening. But it turns out that if you look at that same area and you're simply looking at how it's modulated with the word rotation, then the latency only shifts and not the width. And so it seems like the onset becomes longer with the different rotations, meaning that it's probably maybe a downstream effect from the other part of the brain that's doing the rotation, feeding it into the Broca's area, and then you finally have the effect. So this is a hard experiment to do, and it's hard to interpret, but it's still a potential for pushing out these more subtle resolution or pulling out these more subtle effects based on the latencies. Okay, so in an ideal world, let's say there was no hemodynamic effects whatsoever. Let's say the brain behaved perfectly. It was all just this mesh of capillaries. Given the signal to noise that we have, I mean, we're up against a limit of signal to noise, and we're being generous here. Let's say we have a 1% noise, a 4% bold effect, and we have 256 time points per run and a 1 second TR. So, and let's say we're trying to then push the resolution of our experiment with this data. It turns out that just because of the noise, it would take us. So if we were doing a paradigm in which we had a block design of 16 seconds versus 16 seconds on versus 16 seconds off, it would take us 11 runs. I'm sorry, it would take us 21 runs to get down to a temporal resolution of 100 milliseconds to discern the difference between 100 milliseconds. If we had a more optimized paradigm, and this happens to be 8 seconds on and 8 seconds off, where it looks more like a sine wave, then it would take us about 11 runs to get down to 100 millisecond time differentiation. And this is purely due to the intrinsic signal to noise of the signal. So in an ideal world, that's sort of our upper limit. I mean, to push a 100 millisecond resolution, it would take us 11 runs, and that's even given the fact that if there's no hemodynamic effects that are smearing things. Okay, the last way of pushing temporal resolution is another sort of clever paradigm approach that was developed by Seiji Ogawa, and that is relying on the fact that different parts of the brain inhibit other parts of the brain. So it turns out that if that happens, you can actually map out the speed of transmittal of the inhibitory effect. So in this case, he actually had forepaw stimulation. And it turns out with mouse forepaw, if you stimulate one forepaw, it will inhibit the signal from the other forepaw. And if you stimulate that as well. So you stimulate two, but if you stimulate them at just the right timing, then the inhibitory effect will show an effect. So here's an example of sensory cortex 1 or 2 versus sensory cortex 1. So if you actually have a 100 millisecond delay between stimulating one forepaw and the other, you'll get no inhibitory effect. It'll be a perfectly high amplitude signal on the other forepaw. But it turns out if you wait until about 20 to 50 milliseconds of the delay between the stimulation, you get a maximum inhibitory effect of one forepaw and the other forepaw. And that will be manifest as a slow, sluggish, bold signal change, but you're just looking at the magnitude with this short delays. And so you can actually see this dip in bold magnitude as a function of this very, very rapid or very brief signal delay. So that's one other way of maybe looking at very, very rapid network communications that's inhibitory. The key is that either has to be excitatory or inhibitory. You can probably look at one or the other with this timing modulations potentially. Okay, lastly, sensitivity. Not too much I really want to say here other than we're trying very hard to increase our sensitivity. We're always up against signal to noise problems. Our contrast to noise in functional MRI is anywhere from 4 or 5 down to less than 1. So it's a very narrow window in which we actually pull as much signal out as possible. And one of the ways of increasing sensitivity is increasing field strength. And it seems that so far, and maybe this is starting to taper off right now actually, is that, yeah, it definitely is. There was a linear progression of field strength, of human field strength versus year. But now it's at 11.7 Tesla, it's going to taper very quickly. Another way of increasing sensitivity is by adding RF coils. So if you have multiple RF coils that are smaller, they see a smaller area, but their signal, their sensitivity is greater. So the idea then is that that's why we have multi-channel coils now, is that you can have, that's one of the reasons. Another one is to helping in spatial encoding. But for signal to noise, you can have up to 16 channels. Now we have up to 32 or 64 channels. That basically is 34 little flashlights in the brain essentially that it's much more sensitive than just simply having one quad coil that we used to have in the 90s. So, okay, but there's one caveat to that that's really important to know as well, and that is you can have a signal to noise of 1,000 to 1, and unfortunately, with what matters in fMRI is your temporal signal to noise. So each image could have a signal to noise of 1,000 to 1, but if you're collecting time series data, your signal to noise isn't going to get much better than 100 to 1, okay? And that is because of the fact that, so this is temporal signal to noise versus image signal to noise. And if you're looking at a bottle of water that's not alive, it's a 1-to-1 proportion essentially. You have, if you go up in image signal to noise, if you collect a time series of those images of a bottle of water, you'll have also, if you have 1,000 to 1 image signal noise, you'll have 1,001 temporal signal noise, theoretically. But if you're imaging a person, though, because of physiologic noise, because of breathing and the heart rate and bold fluctuations, which are interesting, which actually are the good sort of noise, but it's only a fraction of the signal, those all interfere. And so, for instance, CSF, which is the most noisy because the CSF sloshes around the brain, your limit is about 50 to 1. Even if you have your images that you collect it with have a signal to noise of 400 to 1. So the optimal place to be is probably at where this image signal to noise equals the temporal signal to noise. So with gray matter, it's about 100 to 1. So if your image signal to noise, you want to then decrease or increase your resolution until you have lower signal to noise, because the signal to noise is proportional to the voxel size. Make a larger voxel, you have higher signal to noise. Lower voxel, lower signal to noise. It's directly proportional to the voxel volume. And if you have a lower signal to noise, you can increase your resolution such that the image signal to noise is about 100 to 1. And that's probably the best place to be for doing functional imaging, because that's where you're making the most of your resolution as well. Okay, as I mentioned, there's all kinds of, and also I want to emphasize is that, we're all working very, very hard, because I think all of this noise that we have here is very filterable. We can get rid of it if we understand it better. And we're working very hard to get rid of it such that we can start to move this curve more towards this, but we're not quite there yet. Okay, and then you can keep on, then you can get much more sensitivity. So a lot of things go into fluctuations: CSF pulsation, vasomotion, breathing, bulk motion, scanner instabilities, changes in blood CO2 with breathing, and then finally what we're interested in, looking at resting state fMRI, which I'm not talking about at all today, is spontaneous neural activity. I think I'm going to pass on this, given that we're at the end. I just want to end with this one this one point, and that is that if you do average enough, there's more signal there. This is a work by a person in my group, Javier Gonzalez-Casillo. He did this study where he just averaged the heck out of the data. He averaged for 9 hours and did a block design, a very simple task of tapping their fingers when they saw either a number or a letter, just to keep their attention. And like I said, he averaged like crazy. And he did one other thing too. He didn't confine it to having a simple model. So typically we just do a sustained response model. That's our model of the signal change. Sometimes we can put in other models, like a transient effect at the very beginning and the very end. This is something that might happen neuronally that most people don't really do. And then the third model is what we have is basically an unconstrained model. It's essentially a SHA function that captures anything that is repeatable, no matter what the shape. So he did that, and with this data, it's hard to make sense of it unless you cluster the signal changes that look like each other. Okay, after averaging for 9 hours with that very, very simple task, he found this, which essentially was all of gray matter becomes activated or deactivated in some way. So you have your canonical responses that you expect to see in the visual cortex and the motor cortex, because it was a visual motor task, but you also see all kinds of other stuff, like you see these massive decreases. You see some transient increases at the beginning and end. You see, a decrease and an increase, all kinds of things. So you have all these signals that we never even expected to see that show up. And after this much averaging, it's pretty much the whole brain that does something. Okay, yeah, and so that basically is showing that there is more signal there to be had. We'd like to not have to average for 9 hours to do it. And so our goal is either to use temporal filtering or higher field strength or other techniques to sort of get at that goal. But so we still have a long ways to go. All right, thank you. Okay, any questions? Yeah.
So for just a clarification, early on in the temporal resolution slides, where you had the different response functions where you're saying how short of presentation?
Yeah.
What was the task that you did?
Oh, that was just simple visual simulation.
Okay.
That was a simple checkerboard.
Okay. So then if you're doing some kind of event-related
Well, no, it will still be a gamma variate function. It's just that the amplitude of the gamma, I mean, it's actually a good thing. For very short, I had a few other slides that sort of helped clarify this, but I skipped them. For a very short stimulation, you still get a gamma variate. It's just that the amplitude is larger than you would expect from a linear system. So linear system, you model this, if you give a very short stimulation duration, like half a second, you'd almost have nothing left. You would barely see it. But typically, when we do event-related, if you give a half-second stimulation, you get a pretty nice big response that looks like a gamma variate. It's just bigger than you would expect for the most part. That's all. And that's usually a good thing. Most people don't really mind, unless they really care about looking at, the amplitude of bold as a function of duration to sort of get at what neuroactivity is actually going on. And then you have a problem. So even if you look back at Logothetis' paper, 2001, where he was actually systematically varying contrast, this wasn't duration, but it was contrast versus magnitude, you get a nice relationship. But down to very, very low contrast, it becomes extremely nonlinear. So it turns out, it seems that bold contrast tends to amplify a little bit, either low contrast or any sort of low-level neuroactivity seems to trigger bold in a way that is nonlinear at very low contrast or very low durations. So, okay. All right, well, thanks. Thank you. Great. And the next class we're going to skip then this week, and then I think the next one is next Monday at 2 o'clock. I think most of them are going to be in this room. And and then we'll just do 3 a week spaced out that way.